Hello everyone, this is Dr. Ashfaq Ahmed working in the field of structural biology and bioinformatics. First of all, I'm thankful to all the viewers for watching my tutorials and lecture videos. Today, I'm going to introduce the latest version of Boltz-1 that is called Boltz-1x. It is running under the MIT license and is developed by the MIT team. Before, we had covered the tutorial for Boards 1, but now its new version is released almost a week ago. So now I'm going to perform a tutorial video for you on Boards 1x. What we will cover in this video. First, I will cover the tutorial for modeling and docking. And in my tutorial, I will try to incorporate two different ligands. And that's why you will learn like how you can test many ligands or more than one ligand in a single run. And in the middle, we will also compare the differences between boards 1x and boards 1 and the comparison of boards 1 with and boards 1x with all others top of the line programs available so far. So first of all, let's come to the tutorial section. First of all, this tutorial I brought for you is on Google Colab. So you have to type this link in your browser. If your internet has some issues or if you are not able to see it clearly, then follow the description section of this video. You will follow this link. And once you enter it, you will see this kind of window. You can see. Now click here and select this one. ASH100/DAS. Once you click here, you will see Boards 1 structure prediction tool. That was the previous one I had covered before. Today we are going to cover Boards 1x for biological modeling. And now click here. Once you click here, you will see a short description of myself, and then for the video tutorial or for this tutorial you will see a link. In case you are using this program right now, then you have to cite these two papers. Once you click here, it will lead you to those two citations. Particular board 1x citation is not released, so once it is released I will update it here. Okay, now you have to click here, connect. It may take almost 20 to 30 seconds for your connection. So let's see. Now it is connected. Okay. Here you have to paste your protein sequence. I'm pasting this one, but in case you have more than one chain, let's say you have chain A and chain B, in that case, first paste the chain 1 sequence, then you can provide this colon and then paste another chain, like let's say this is my chain B, or in case I would say it's a dimer. Then can you see? So you have to separate both the chains on this colon, and for other complexes, like whatever how many complexes you have, so you can do that way. You can incorporate one, two, three, or as much chain you want to include. So in the current tutorial, I will include only this short kind of peptide. And if you have a ligand input, then you need to paste it here. From where the ligand input will come, let's say you have a pumpkin. Let me open. So, for example, you are using, let's say you have paracetamol. Okay, so now acetaminophen, this is, for example, this is your ligand. So you will see smileys here. Oh no, came here. Come here, wait. Okay, it's here. You can see here the smileys. So copy, and if you have more than two, so this is the one, then separated by colon, and it will be another smiley. That's it. But right, I will show you how you can incorporate two smileys or two ligands. One from this window, ligand input. I'm using this one. Actually, you can search it like just drop the smileys into the pumpkin. It will tell you it's L-tyrosine. So I'm using one L-tyrosine, but I'm also using another ligand. Its name is SAH. That is called CCD of the ligand. So in case you don't have this and you are working on some another database, not a PubChem, then you want to use CCD. So you can also incorporate CCD. Okay, from where we can pick this CCD? Let's say I'm working in the protein database. Okay, it's open here. So you can just mention here name of your ligand. Let's say I'm mentioning SAH, or you can just write the name of your ligand. It will provide you the CCD. So this is the actual ligand I'm using, and this one is called SAH, S-adenosyl homocysteine. So I'm using this one SAH. So, for your understanding, here is your protein sequence with one chain or two chain. Here is your ligand input, one, two, three, four, whatever. Or if you want to provide the SH, like from the CCD name, not from the smiley, so you have to provide here. If you have more ligand, let's say one is ZX8, for example, this one. So normally also once you have a PDB structure and you open it in your any visualizer of your choice, Chimera, PyMOL, Discovery, they also show this name of the ligand SAHZXE1L2, whatever. Okay, so in this tutorial, our program will first model this one, then we'll incorporate this ligand, L-tyrosine, and this one is the S-adenosyl homocysteine, this one. Okay, if you want to use some common name, but it may lead to some problem at some time, so I would not suggest to use the common name because the common names are at times they are dependent on the caps lock and maybe some spacer or the spelling. So I would suggest why not you can use uh, either CCD or either the smileys. It's up to you. Okay. If you are using a DNA, so just provide the DNA sequence here. Okay. And if you want to name, let's say I name it tutorial_1. So that is up to you. Once you click here, you will see this kind of option. I would suggest just click run anyway. Now you can see uh, the notebook has captured or has picked this information. Now here we will install the Boltz-1 and some other dependencies required to run this tutorial. So we will click here. It may take 3 to 4 minutes depending on your internet connection. So far, while it is running, I'm coming back to these slides and cover this part, like what is the difference between Boltz-1 and Boltz-1x and with other programs. So remember, in Boltz-1x, they have incorporated inference time steering. That is also called board steering. What actually board steering is? Board steering is actually inspired from the Feynman cake diffusion steering. You can search about it because the tutorial is not about to introduce or to talk about the Feynman cake diffusion steering, although in my other videos I talked about the RF diffusion. I did not talk about the Feynman. So you can have the idea about the diffusion and all those kind of things from the literature. Okay, so in boards one, they have introduced board steering, which is actually inspired from the Feynman cake diffusion steering. What it does, it actually enhance the physical quality of the model. At times, you may, if you are using these AlphaFold 2, 3, Protein-Ix and Chi1, if you are using these kinds of things, then you may experience sometime that the generated model contains so many chirality problems or steric clashes. Uh, but because of this update, uh, it is high likely that your model will be clash free. It will not have any distorted geometries, wrong chirality, or steric clashes or overlapping chains. So this board steering in boards 1x actually enhance the physical quality of the structure. Now, Board 1x also using gradient step for resampling under the force field inspired potential to define prediction using diffusion. That also significantly improve the pose accuracy. To simply explain this step, it means that when you are using a multi-domain protein or a multi-chain protein or a protein with certain ligands or a protein with a DNA or RNA, whatever, so the the bold X will use a gradient step, just like it will generate so many models in first phase, then it will resample based on energies. The information will be coded in the force field. So using the information from the force field program will just like gradiently will filter out and will signifies or will bring you the most the best model, and that's why it will increase pose accuracy between different chains. The chains could be of DNA or the chains could be of compounds or proteins. Okay, and the third thing which is incorporated in board 1x, that is the cyclic support for different peptides and for different cyclic amino acid and compounds. So it will also enhance the accuracy. So that was the critical or the significant three improvements brought in boards 1x. They have also tested the performance of boards uh, 1x on PDB datasets, which had the accuracy or the confidence CI means confidence interval, which had the confidence interval of 95% already. So they test with AlphaFold 3, they test with High 1, they test with the older version Boards 1, and they test with uh, Boards 1x. And then they perform for mean LDDT. I have explained this in detail in my AlphaFold video with the doc Q score and the mean LDDT and the RMSD, but and the physical validity, which I explained in my earlier slide. What does it mean, the physical validity? So if you see in all other things, they are comparable. The Boltz-1x or the Boltz-1, it's comparable with other programs with slight differences, but the physical validity range or the clashes things, it exceeds, it's left the Boltz-1x left other programs far behind. You can see this one is the AlphaFold 3 and this one is also AlphaFold 3. The next one are the Chi 1 and the older version Boltz-1 and the new version Boltz-1x. So I suggest if you are at the beginner level for modeling or docking, and if you don't know which program to use, so you can consider the Boltz-1x. And this picture is taken from the GitHub, and this is the address. You can go there and check your set. This is their official repository for the boards group of the boards group. Okay, so that was all about the differences of boards. So let's coming back to the original tutorial section now. This is done. It took almost 2 minutes and 38 seconds. Now we will click this option and it will generate the multiple sequence alignment using the Colab. Okay. And it took almost 8 seconds because my protein was too shorter and too known. So that's why it took 8 seconds. In your case, it may take almost a minute or so. It depends upon your target. Okay, now here we are going to predict the structure using Boltz-1x. I will correct this. But that's okay. Boards, because the code I have changed it, so it's not now. Let me correct it now. One. Okay. So let's wait for 1 or 2 minutes. In the meanwhile, we will talk, or I will talk to you. Okay. So now it's 32 seconds. Okay. It will not be so longer. Okay, dear viewer, if you haven't subscribed this channel, I request you to subscribe, share with your colleagues. Um, I'm trying my level best to bring the exciting and the new stuffs from the field to you guys. I also know many of you guys have requested for so many different tutorials I have in mind, but the problem is I'm also managing my time. So in near future, I'm trying my best to give you all those tutorial in one day, but this is not possible. I'm also human. So just stay connected, share this channel, these information that will boost me, that will boost my energy to bring more exciting stuff for you. And I'm also thankful for these 3,000 and so much, 3,000 plus subscribers I have. I'm thankful, extremely thankful for your support, whatever you are. Stay happy. Okay, after this step, we will download the result and I will quickly open in PyMOL and I will show it to you that how things are working, what we have done, what we have achieved, whatever. So right now, I think it may take few more minutes. So that's why I'm going. Okay, it's done. It took 2 minutes. Otherwise, I was trying to pause it. Okay. Now you have to click here and it will download results underscore tutorial underscore one into my download folder. Here is my download folder, this one. Okay, let me create a new directory. This one will be TT, let's say, and keep it here. Now open this and let me extract here. Now, you have four files. One is the MMCIF file. Most of the guys are asking me that we don't have the PDB file. Actually, this is the PDB file, but this is the protein model file, but in CIF format. So once you open it in your Discovery Studio or PyMOL or Chimera or some other program, just click Save As and save as a PDB. It will be okay. This one you can open the YAML file in the right click and open it with the notepad. You may have all those information if you want to use it in your thesis or things that what we did, how we did, what program used, how many recycle steps, whatever, blah blah, so you can use it. This is the picture from the MSA we perform in the earlier step, that prediction. So in this step, you can see it tells you that for this position, we have almost 58 amino acid protein, and it, it captured almost 8,000 sequence for this, and then the number became decreased, but that is fine. Uh, and this is the A3M format for it contains the multiple sequence alignment in A3M or the A3M file. Normally, the bioinformatics people, we are using the faster format or all those things, but with the way we generated the Colab fold, the MSA, the Colab fold will save your file into the E3M format. Okay, so now I'm going to open how to say. Okay, let me open the PyMOL. Oh my God. Okay, now uh, this is my PyMOL. So let's go to the download this TT, and then from the TT you will see this MMCIF file. So to visualize it more, let's say display sequence and change the color background to white. Okay, make it bigger. Okay, so now you can see here it has two ligand. In chain C is SAH, this one is a SAH, and in chain B I have the LIG. The LIG is basically the L-tyrosine. I use the smileys, and this one is basically the SAH. So why I use two two ligand, because I wanted to show you that how can you incorporate more than one ligands in a single experiment. But I also you can incorporate more than one ligand using the smileys, uh, using this one. Let me come here, using this one. But uh, what I did, I use one here and one here. But that is up to you. If you want to take the smileys of SAH from somewhere, from PubMed, and then use the colon and paste it here and leave this blank, that will also be fine. So that is up to you what to do. So I hope you like this tutorial. If you have any suggestion for improvement, you can ask me, or you can drop in the comment section. Thank you so much for watching me. Stay happy. Bye bye.